Cool, thank you. All right. Good afternoon, everyone, and welcome to this week's Earth and Environment Graduate Seminar. My name is Kevin. Presenting today, or helping me present today, is Connor Wolf and Holly Mulligan. Today's speaker comes to us from the University of Florida. He works at the Institute of Food and Agricultural Sciences and his specializations in soil and water nutrient management, and are working towards sustainable agriculture. He has served on numerous scientific review boards, including the National Science Foundation and the USDA. He had a specialization early on in geology, but later got his PhD in soil and water sciences from the University of Florida in 2009. So thank you everyone for joining us. And without further ado, please welcome Dr. Bhadha.
Good afternoon, everyone, and thank you so much. I want to first extend my thanks to Dr. Bhadha and Dr. Sridhar. Thank you so much for the invitation. I was just had a lovely lunch with a group of students. I was just letting them know it was probably 18 years ago that I visited FIU campus. We were grad students, just like you all here in the room. I was at Gainesville, University of Florida. I had friends here at FIU, so I used to come down and visit. A lot has changed since then. Anywho, someday you guys might be in this position giving a presentation. I'm going to talk about integrated soil, water, and nutrient management. And this is pretty much what my program is all about. And personally, I feel these three aspects are pillars for sustainable agriculture. So, like I said, I think soil sustainability, soil health is one component which is really critical for sustainable agriculture, agroecosystems. Water quality and its resource is a second pillar that I think is essential, and I'll be talking about that. And the third, and definitely by no means the least, is nutrient management, which, you know, a lot of times we would understand it as fertilizer management, the different fertilizers, your nitrogen, potassium, phosphorus, some of the micros that you add into the system. So taking an integrated approach of all these three aspects really is pushing the sustainable ag pendulum forward. Let's talk about soils for a few minutes. Why is soil sustainability so important? Because almost 20 plus billion tons of topsoil is lost globally. And just in the US, we've got about 1.7 billion tons of soil that we lose annually. The biggest culprit is wind erosion. A lot of times, if you are working in the ag sector, in the month of April, May, it gets really dry. The soil is fragmented, dusty. And if you have a huge wind that passes through, you're going to lose a big chunk of that topsoil into your ditches and your canals. Runoff, water erosion is a second big factor for loss of topsoil. And then there is a third factor, which is kind of near and dear to myself. Here in South Florida, in the Everglades agricultural area, is the soil loss of organic histosols to a process that we call oxidation or soil subsidence. I mentioned the Everglades agricultural area here in South Florida. It's just south of Lake Okeechobee. It's about 500,000 acres of farmland. The predominant crop grown there is sugarcane. Almost 50% of US sugar comes from this region, only a few miles north of y'all. It's comprised of histosols. These are organic soils. Underneath these soils, there is a limestone bedrock. Limestone, high pH. So there are a lot of reasons why these soils are important to understand because they have high pH. A lot of our micronutrients, even our essential nutrients, get bound or non-bioavailable because of this high pH. The area was drained back about 100 years ago to make them more adaptable for farming. And since we started to drain this organic landscape, we have exposed these soils, these organic soils, to the atmosphere, and we are losing them almost a centimeter a year through a process known as soil subsidence. Let me tell you, this soil that my colleague's holding in his hand has more organic matter than the compost you would buy at Lowe's or Home Depot. It's a lot more nutritional than what you would find in your big stores. So why is soil health important? There is a whole list of reasons, and I'm sure Dr. Mahadev and Sridhar have discussed this as part of your agro systems class, like less weed pressure, increased diversity, soil stability. There are the two that I've highlighted at the bottom there: increased soil carbon and increased organic matter. Those are really, really critical, especially in the times that our farmers and our growers and us as scientists are working in today's condition, especially with carbon sequestration and carbon credits and carbon markets. So that's why that makes it really, really interesting. And then those that are learning about soils for the very first time, well, just a little note. Did you know that over 1 million microbes and over 50,000 different species of bacteria can be present in 1 gram of soil? So soils actually are quite a mixed bag of interesting things going on. Why is soil carbon so important, right? It's important because it is the foundation, it is the keystone for all physical, chemical, and biological processes and properties that help a soil to be healthy and nutritious. As you can see, carbon right there in the middle, you've got all these other different nutrients, these different elements, they all bind themselves to that carbon molecule. So it is important for us to address this carbon issue. How have I gone about pushing my program forward, trying to understand soil health? So this is just the framework. Again, there are different frameworks of how you can push the science of soil health forward. This is something that we try to apply with our stakeholders, with our growers. So regardless of what cover crops or soil sustainable practice that you might have, we try to identify fields where there is some treatment going on and compare it with a control site. We will do pre and post assessment of soil samples from those field sites, bring those samples back into the labs. I was happy to see some labs where there is some work going on, and we test the whole host of soil health indicators. The Soil Health Institute has come out with their 17 list of phase 1 indicators. They're adding some more indicators as they go along, and we try to mimic those in our lab. And then we take all that pre-post information back to our growers, back to our stakeholders in the form of outreach and extension. So that's how we kind of push that science forward. When we first started in this space, we only were able to do a handful of soil physical analyses in our lab, but over the last decade or so, we've been able to kind of add a whole host of series of soil health indicators in our lab that help us develop soil health indices ultimately. I was asked earlier during lunch, can you shed some light on soil microbes and soil biology? How important is that? Very, very important. Historically, the soil scientists worked on soil physics and developed those fundamentals. Soil chemistry, even today, soil chemistry is the meat and bones of any soil study. But I would say in the last couple of decades, and more so even now, soil biology, microbiology is trumping both soil physics and soil chemistry. It is very, very critical to understand soil microbes as well. So here's a applied example of how we go about doing things. Obviously, as faculty researchers, we want to get some funding. So we receive funds from the Southern STARE program. And what we decided to do was evaluate some of the cover cropping practices that these growers across the state conduct during the fallow period. For us here in South Florida, summer is typically the fallow period. It's really, really hot for us to grow any row crops, any cash crops. Whereas if you go further north or even in the panhandle, believe it or not, it's the winter time. That's their fallow period. It gets really cold and they just can't grow a whole host of cash crops then. So we identify these growers, we go up to approach them and say, hey, can we do this soil health assessment in your field at no cost to you? We are not asking for any financial from you. And we will provide that information back to you so that you can use it and incorporate it in your management practices. And here's an example of results, how we take that information back to our growers. And this is typical of what extension would look like. Extension really is not about heavy statistics, heavy scientific information. It is breaking down that science so that our end users can understand. So 6 out of 9 growers were able to show that there was an increase in malic-3 phosphorus as part of their cover cropping practice. However, just 1 out of 9 grower was able to see an increase in potassium. So we can clearly go back to the grower and say, hey, if you're going to do these cover cropping practices over this period of time, you want to make sure you apply a little bit of potassium right after that for your cash crop, because we've seen a reduction in potassium. So that's one way that we approach this. We take some of this preliminary results and go for a larger initiative, a larger project. And in that project, we kind of tend to do the same type of work, but we end up adding some of this biological and this microbial work, which is probably a lot more convoluted, but just as important as the soil chemistry and the soil physics is concerned. Some results obviously shown up there that if you were to do some cover cropping practice, you do see some significant increase in your water holding capacity, right? So let's say if you've conducted some cover crop, some sun hemp, some cowpea in your field over the fallow period, once you've harvested that cover crop, and if you measure the soil samples, you'll see its water holding capacity is a lot higher. So during a drought period, a prolonged drought, that excess water holding capacity can be beneficial because there is still some moisture left in the soil. And growers can relate to that language. They really, you're going to lose them if you're going to start talking about a whole lot of statistics and methodology of how you capture that. We also capture some anecdotal information, so to speak, having discussed with surveys with our stakeholders, with our growers. And that qualitative information is just as important as the quantitative work we do in the lab. Here's another example of an interesting topic that we worked on, again, important from agroecosystems point of view, the application of soil amendments. Any sort of organic waste product really doesn't need to go back into the landfill. We need to be creative as researchers to try to find ways how we can put that feedstock back into the field, back into the system, and investigate that. This is exactly what we were doing with US sugar. You know, when you harvest sugarcane, there is a byproduct called bagasse that is generated. And just here in South Florida, across the 3 sugarcane companies, almost 3 million tons of bagasse is generated annually. And I've seen that visually. It's 3 football field size of material that is generated annually. So it is clearly not a non-renewable resource. So that's attractive. We're getting this material every year. What can we do with this feedstock? When we looked at it nutritionally in the lab, just at the material, we realized there is quite a bit of micronutrients present in it in the first place. So even if you were to apply it back into a depleted soil, you are potentially going to see some effect of those nutrients being plant available and bioavailable back to your cash crop. So here are some images of a study that we conducted. That's that huge acreage of bagasse through an aerial image, and then its application into the field. Believe it or not, the biggest constraint for a grower to apply material back into the field is logistics. The distance from where that material is generated to where we have to put it back in the field. So there is a huge investment from growers just from transporting any material. So the closer you are to your feedstock, the closer you are to where that material is generated or produced, the better it is from a cost assessment perspective. Yes, Mike?
This brings a question to mind. So what is it exactly? Is it squeezed? Is this what's left over after squeezing the sugar cane?
Sure. What is bagasse, right? What is this cellulosic material? It is the material that is pressed and squeezed. It is cane that is pressed and squeezed. There is another material that is also generated, and that's molasses. So there is a lot of liquid material that's also generated, and that tends to have a much higher sucrose content. But bagasse is just fibrous. It is just cellulosic. It is material that is squeezed. There is another product from sugarcane mill that is generated. It's called mill ash. That's another product. It's more charish. It's ashy material. And a lot of work is done in mixing ash with bagasse and applying it back into the field. And that is what you are seeing here as a study where US Sugar, instead of using and incinerating all of this material, what they were doing to generate energy as cogeneration, now thinking about putting it back into the field from closing that loop from a sustainable ag perspective. So that's when we come into the picture and they are like, well, if we are going to do that, might as well get some researchers to see what's the effect of applying this material. And we didn't know what we were getting into. And so we did a long-term study, and just to kind of highlight certain aspects here, it's not a one-to-one direct effect. It's not like if I had a bucket of bagasse and I planted it into the field, wow, my cane is going to grow great. As a matter of fact, what we've seen that in the first couple of years, it's really not that effective at all, because this is cellulosic material. This is material that has to break down, that has to be biodegraded for the plants to actually take that up. So the growers actually had to apply a few units extra of fertilizer just to get that plant cane growing. However, in subsequent years, once that cellulosic material broke down, we were seeing higher numbers of malic phosphorus. You've got some phosphorus in the soil that is being liberated because of that bagasse that is breaking down. So keep in mind, a lot of these processes are not directly related to responses from the get-go.
Is it the nutrients or the texture of the soil that's providing the advantage?
It is the nutrient.
It's
It is the nutrient, right? It is not so much so the composition, not so much so the texture. Surely the soil has more dirt to it and a little more texture to it, but it's the nutrients that's getting released. Okay, so the EA, that is where we are doing most of our work. That green area, light green area shown there. A lot of you all have worked in with water quality and are aware of the water conservation areas, the SPA, the stormwater area, Lake Okeechobee right there. So just to give that little synopsis, almost 450,000 acres of organic soil, 80% of that is used for growing sugarcane. Florida topography, super flat, right? Less than 1% gradient here. Typically impermeable bedrock, our marl, our limestone. So that has a lot to do with hydrology here. You've got this impermeable limestone underneath, so the water table tends to be pretty high. Soils have been actively drained by pumping water through these systems so that we can facilitate farms. And the water leaving the EA are routed through into those dark green STAs, stormwater treatment areas. These are man-made wetlands that further treat the water before they make its way into the water conservation area and then down to the Florida Everglades. Irrigation and drainage, again, are very unique for the EAA. It is bottom top. Irrigation is bottom top. You are not going to see irrigation boom lines across the fields feeding the fields. So it's a network of ditches and canals. It is all lateral subsurface hydrology, and that's managed through the networks of ditches and canal. So what you are seeing in that bottom image there is a typical farm canal with a pump house. Here you see three pump house. When the farm wants to bring the water in for irrigation, the pumps are turned on, the water gets into the main canal and makes its way into the ditches. From those ditches, subsurface water raises into the fields, and that's how the fields are fed. So it's unique. You're not going to see overhead irrigation lines or tile drainage either. With farming, I did mention 80% of farming is sugarcane in the region, right? So let's think about farming over a 4-year period. When you talk about sugarcane, it's typically a 4-year cycle. October, this month, most of the planting will start. This time of the year, planting starts. So let's say you were to plant cane, that's how it's planted, the stalks in rows. In the month of October, November, planting can go on all the way into April. Once this cane is planted, it takes about 13 to 15 months for this cane to kind of grow up and get harvested. Once that cane is harvested, there'll be another crop that will emerge from that same plant, and we'll call it first ratoon. That might take another year, 13 months. Once it's harvested, once it's grown, it gets harvested again, and a second ratoon grows from it. Depending when that second ratoon gets harvested, typically in the 4th year, there is potential to go in and plant some specialty crops on that same plot of land. So there's sweet corn, lettuce, beans, radishes, cabbage, celery, all those crops are grown in the EAA. However, if it's the warmer time of the year, April, as a matter of fact, even sometimes late March, April, May, definitely June, July, August, there is room for us to flood these same fields and grow flooded rice, cultivate rice. Did you know almost 25,000 acres of rice is grown in our backyard in the EAA? But it's not so much so grown because of its cash value as much as for its environmental and cultural value, so that we can slow down that oxidation of organic matter, organic soil. Lots of benefits of growing flooded rice in the EAA. I've touched on a few. Almost 50,000 acres of sugarcane land is potentially fallow in the summertime in South Florida, right? So we've got 50,000 acres of land fallow. Why should we leave it exposed to the atmosphere and lose all that organic matter, lose all that carbon? And that's when the growers have stepped in and said, let's flood it and let's grow rice. Again, 50,000 acre fallow, 25,000 acres are planted. We've still got a long way to go. That's only about half of it. We have potential to grow more rice in the region. But what was really, really interesting to me was when I look at national rice cultivation versus a small little Florida, no NPK applied to grow rice in the EAA. Boy, I should make a t-shirt and wear it. Arkansas grows 1.4, 1.5 million acres of rice, #1 state in the US. California, Texas, Louisiana, they all grow rice. Florida, 25,000 acres, we are less than 1% of the rice footprint in the nation. But we do not apply any NPK. We are using the same starter fertilizers that are either in the soil from the previous cash crop or utilizing what's in our muck soil. So when we take this concept for USDA funding, NRCS conservation folks, and we put it up front there, wow, no nutrients applied and you're still cultivating a cash crop, must be something going on there. So that's where NRCS comes into play and provides us with some funding. This is an interesting call. It's called SEEP, Conservation Effects Assessment Projects. Across the US, there are about 30 different SEEP sites that are working towards understanding different conservation practices, but quantifying those so that they can develop models that folks like Dr. Sukop and others can use, right? And so we ended up doing one of these projects where we are looking at the effect of flooded rice in rotation with sugarcane to see if there are benefits to soil and water quality. So that gave us a really good niche a project statement, a research gap to actually be able to quantify are there benefits of doing this rotation. Again, what I wanted to show here was the importance of this concept of subsurface flow. It is very unique, yet extremely critical for how water flows through our 500,000 acres of farmlands in the year. It is lateral, it is subsurface. So it's very important to be monitoring groundwater more so than your surface, what's in the surface water, because a big component of that is just lateral movement. So then obviously, as far as the monitoring work goes, postdoc students installing all these instruments in the ditches and canals. And then, presenting this information. So let me highlight another interesting item here. Let's see. All right, right here. This is the famous subsidence post. I don't know if some of y'all might have visited the EREC in Belgrade. This was a 9-foot concrete post that was inserted in the ground back in 1924. If I was standing here in 1924, I'd have been standing right there. And we've lost 6 feet of organic soil, that muck soil. This is a post only about 100 yards from my office in Belgrade. We've got about 3 feet of soil left remaining here. No one went in there and excavated that soil out. Natural loss of organic peat soils to the atmosphere. So conservation practices in this region is really, really important to conserve that remaining 3 feet of soil in the region. Grad students, postdoc obviously go out and do their magic, right? And then here it is, we are presenting their efforts. But what I wanted to show is the variability of soil in the region. It can range from a few centimeters, inches, all the way to a few feet. So it's not consistent. I can't stand here and say we've got about 2.6 feet of soils remaining. No, sir, no, ma'am. In some areas, we've only got a few inches, and there's limestone bedrock, and in certain areas, we've got a few feet of muck soil still there. But what we found was that even though we were losing the soil, even though the soils are getting shallow, there really wasn't any crop response. The yields were not quite being impacted. Why is that so? And that's because of the BMP program, the best management practice programs that come into place in advising the growers on what they should be doing with their shallow soils. So things like soil testing, things like a banded application of fertilizers versus broadcasting. Those are things that allow breeding programs for the different varieties. Plant this variety and not that variety, because they are more stronger to flooding or diseases. So those are things that allow us to combat losses in yields, and therefore we don't see a whole lot of relationship between depth and yields. Gonna shift gears a little bit here now. Okay, so welcome to STEPS, STEPS, Science and Technologies for Phosphorus Sustainability. STEPS is a NSF funded STC where we are doing all sorts of research related to just phosphorus, right? And again, at lunch, I had a really good conversation with the group about there's a whole team of folks working on carbon sequestration. I tell you what, there are a whole host of folks around the world just working on phosphorus as well. So hopefully the next few slides will highlight why that is important. STEPS National Center. But what is interesting about STEPS is that it's got 3 unique triple bottom line areas, one in South Florida, one in North Carolina, one in Arizona, where we are not only doing research, but we are doing research along with socioeconomic aspects of doing that research is. So it's important to kind of understand what are those changes as well. STEPS is a convergence research center with phosphorus sustainability as its vehicle. So what is convergence research? You know, back in my days, 20 years ago when I was a grad student, we were asked to become specialists. Focus in your silo, go into your lab, do that and present the work. It's changed now. Those silos are gone. We want to be doing more convergence work. I want to be doing research where I have no clue what the hydrologist is doing, yet he or she is on my team. I have no clue what the social scientist is doing, yet I want him or her in the same room, and we are going to try to address this problem. And I've just used hydrologists. I'm talking about weed scientists, nutritionists, folks doing genetics, folks doing global modeling. Bring them all, converge them all, and let's try to address that problem. That's what STEPS is about, getting us out of our comfort zone. All living things need phosphorus. It is an essential nutrient for life. But our society's use application can sometimes make that nutrient unsustainable and also detrimental, harmful to nature. Some very interesting points brought up here. You know, phosphorus is mined as a non-renewable resource, right? So there's only a limited amount of phosphorus we've got. We know where it's at. We know how much is there. We need it for our growing plants and crops and our food, but only about 20% of that actually ends up in our diet, actually ends up in our system. Rest of it is lost to the environment. So what can we do to try to prevent that loss, maybe even capture it, and even better if we can reuse it? Okay. Only 2 million tons a year was grown since the 50s, tenfold increase. Now almost 20 million tons a year of phosphate is used for growing plants. Our whole idea about phosphorus utilization used to be single use, extractive process. We mine it, we use it, we lose it. That has to change. That concept has to change. And how are we thinking about changing that? Is by developing interventions in this phosphorus flow diagram. How can we work with convergence teams to try to come up with solutions where we can capture some of that phosphorus and reuse it back into the system? That's Sustainable Ag 101 for you right there. So I'm happy to be working with a whole host of, guess what? There is a group, look at this gray. There's a group of folks out of Arizona that are actually capturing human urine and trying to extract the phosphate and the nitrate from human urine and put it back into the system. All right, so the vision of STEPS, 25 and 25, it's a catchy vision. Facilitate a 25% reduction in human dependence on mined phosphate and a 25% reduction in losses from point to non-point sources over a 25-year period. This is a ridiculously challenging vision. It's a tough ask, but that's why we've taken it on. That's why we are working towards it. STEPS works across 17 orders of length scale. We've got groups of researchers working at the nanoparticle side of things, of how we can capture that phosphorus from our waste streams. We've got folks like myself who are working at the human scale, human technology scale, dealing with farm experiments, farm trials, interacting with our growers and stakeholders. And we've got folks that are working on scaling up at the global scale, at the national scale, modeling, etc. So we've got these 3 different scales working in tandem, trying to address this wicked problem. What do I mean by wicked problem? What I mean by a wicked problem is that if I add a little of phosphorus, I'm not going to be able to see a yield response in my crop. Oops, my crop's not doing good. Let me add some more. Crop's doing great, but guess what? I've made my lake eutrophic. It's a wicked problem because we got to get that balance just right. That's why it's a wicked problem. Anyway, just some faces here of folks that are working on this. And the different projects, yeah, I don't want to go through the whole host of different RFPs that this center is working on, but I'm going to point out on point number 2.5 with a bunch of folks out of North Carolina State. controlling and utilizing legacy phosphorus in our soil. That's something that excites at least myself and the work that we do. So the concept is unlocking the chemistries of legacy phosphorus to maintain sustainable ag and a healthy environment. What do I mean by legacy phosphorus? Legacy phosphorus is that excess phosphorus that is present in your soil profile that has the potential to be lost. A lot of times lost in a detrimental way. So what if we can unlock that phosphorus in a controlled way and make it more plant available, make it more bioavailable, right? So that's the concept that we are trying to wrap our heads around. Wouldn't that be great? We know we have a lot of phosphorus in some of these areas. We know there is excess total phosphorus, but labile P or bioavailable P is very low. So is there an intervention that can make that total, convert that total P into more labile P? And that's what we can sell to our stakeholders. So phosphorus deficiency, more than 40% in our, globally in our arable lands. Fertilizer, huge number that is applied in the field. About 30% is used by crops, 20% of that enters our diet. The rest of it is lost. So there is definitely potential for us to come in here and make that legacy phosphorus as a promising resource to unlock. However, our understanding of this is limited. But that's not a problem. We need to push ourselves to understanding and trying to find ways of making that legacy P more bioavailable. So fortunately, with steps, like I said, if you wanted soils from Arizona, you want soils from North Carolina, now we just pick up the phone, have a Zoom call, get our team in order, and we can do this across diverse landscapes. So we collected all these soil samples from different locations, right? We bring it back into the lab, and we are taking a 4 key approach to unlock that lock of legacy phosphorus. The first one, very quantitative approach using some spectrophotometers, and testing how much total phosphorus is present. Then we can do some conventional fractionation work, Headley fractionation. Maybe some of you all have heard about this. It's a sequential fractionation series where you can extract different forms of phosphorus. And then some specialized technique, NMR or XAS, that we can use to identify the different chemistries of what that phosphorus is within that total phosphorus forms. Different soils, but what I wanted to highlight here, look at that total phosphorus number. That soil has over 1,000 milligrams per kilogram, so over 1,000 ppm of phosphorus in the soil. That's quite a bit of phosphorus in all of these soil types. But look at what's plant available, malic 3 bioavailable. Sure, this one has about 300, about 100, but only about 40 milligrams. So it's a very small fraction that's bioavailable. So now, like I said, where is that intervention that can convert that 1,300 down, or I guess, bring that malic 3 up to 500 or 600, because that's what the plants are going to take up. Different fractionation, this is the Headley fractionation. It's just extractions of different bases, weak acids, and then a strong acid that gives us the different forms of phosphorus in the system. This is what typically is presented in Headley papers and publication. But what I'm showing here is, you see that blue form that says soluble P? That blue form is lacking in pretty much all the samples. That's the plant available. That's the bioavailable P I wish we had in our results, but we don't. And we know that. What are we seeing a whole host of? Here in Florida, soils and sediments, we see a lot of reds and black. That's the calcium, magnesium, phosphorus. That's the residual phosphorus. That's the phosphorus that's not plant available. It stays as legacy P in the system. In Arizona, it's kind of similar. There's a lot more of red. There's a lot more of calcium, magnesium in the system, but very little of that blue labile form or even the yellow form. So Headley provides operationally defined phosphorus fractions. However, It does not provide us the functional group or the chemistry of what that form is. And hence we go into these specialized techniques, NMR and XAS, to be able to do that. So this is a collaboration with a whole host of folks up in Gainesville. And the results, right? What does the results look like? So this is what NMR spectrums look like. And you get peaks of what that compound is, and then you can quantify the area under the peak, and that will tell you exactly how much of monoester bound phosphorus is present, how much orthophosphate is present, how much pyrophosphate might be present. With that information, you can develop your intervention. Because now you know the form of the phosphorus, and there are chemistries that can allow us to release that peak. The same goes with XAS. XAS Zane's work is kind of the same. There are some advantages and disadvantages associated with it. Zane's provides us with a shape based on the standards that we feed into the system. So this is what the results would look like. But from that information, you can make some observations whether there are iron-bound phosphorus is present, whether it's lacking, whether your hydroapatites are in abundance, or if it's lacking. Sodium phytic acid phosphate, is that high, is that low? So that's the kind of information we would get based on our pre-calibrated standards with XAS work. Guess what? It's not just keys 1 through 4. We are starting to push the boundary, keys 5 and 6. I guess Connor asked me earlier, what about microbes? What is the role they play? Bioproducts. I got a grower in North Florida who's working with bioproducts. Yes, there are some solutions. There might be some truths trying to go into looking at some organic acids, looking at some bioproducts to see if those microbes can actually stimulate the soil to make that legacy P more plant available. We don't know. We're going to work on that as we move along. All right, here's a call. We are looking for a postdoc. So folks, feel free to take a image of that. If somebody wants to work in this space, feel free to have them apply for this postdoctoral position. Okay, carbon. Gosh, where do we begin with the carbon conundrum, right? But again, it's very important. It's very important, especially for folks where we are located on muck soils, for us to find ways, how can we slow down that rate of oxidation? How can we conserve that carbon in the system? It's very, very critical. So there are two ways in which we want to approach this. Obviously, the science side of it, the technology factor of it, understanding that, but there is also the social aspect of it. The adoption of whatever technology we use. Are the end users ready to adopt the technology that we are proposing or advising them towards? So it's very important that we are not just too far ahead of what can be adopted. Again, not going to go into the project nitty-gritty, but the concept is to capture the carbon stocks, make awareness for carbon farming and data rights, incorporate carbon farming into our local systems. Every system is different. It's not one glove fits all, and then develop our outreach products from it. Whenever we talk about carbon, it's not just what you see above ground. There is a lot that's going on below ground as well. So taking the approach of above ground and ground, truthing it on the ground, and then below ground is also important. So just highlighting the importance of that. And then when you come to the actual research work, right? How are we going to approach this? How are we going to address this? The workflow objectives kind of help us, allow us to do that. Grad students are going to work across these 5 different steps, look at the microbial activity within the system, look at spectral analyses from above ground, do some machine learning, steps with some other convergent folks, right? Maybe folks like Sridhar who do GIS type of work would be interested. Comparative analyses, and then improve these models and refine them. Again, stealing some of the slides from grad students' research shows the different steps of how we are going to approach this. Ultimately, what is the product, right? What are we going to be able to show to the end user? They don't care about this. A grower, if I had the room full of growers here, they'd be fast asleep by now. Right? But they would be interested to see a map like that at the end, where we are showing how that organic matter is changing based on their practice that they are applying in the field. But it takes a long time and a lot of research to go from here all the way to there. But that's our end goal, basically, develop these maps. Another form of looking at potential solutions to slow down this carbon. And I also want to mention that a lot of things that I'm talking about, EAA, muck soil, South Florida, they are not just unique for us here, right? You can use these same practices, these same philosophies on different systems as well. So that's something to keep in mind. So if you wanted to decrease carbon outputs, you want to start suppressing your microbial organic oxidation. If you want to increase, obviously, the outputs, you don't want to do that. You're going to lose that carbon to the atmosphere. And that's where management comes into place. There are different ways in which we can manage our farms, manage our fields. If you've got a fallow field in the summer, that's what it's probably going to look like. It's going to have some weeds in it. But you can slow down that oxidation by probably flooding that field, converting a system from aerobic to a pseudo anaerobic. It's not quite anaerobic, but you'll slow down that oxidation, you'll slow down that microbial activity. You've got cane growing there, and then you've got a whole host of cover crops that you can apply. Why leave it fallow? Why leave it with weeds when you can go in there and apply cover crops? So all these practices are management practices that we can take back to the grower. Again, the grower would not want to see the results. They don't care about all this data stuff. But what we are seeing is that if you do flood a field, you are slowing down the rate of microbial activity. The enzymatic activity starts to go down. And that is beneficial if you wanted to conserve carbon, because it's your enzymes that's breaking down that carbon. We not only work in row crops and agro systems, but ecological settings kind of are just as important. This is the DeLuca Preserve. DeLuca Preserve is almost 30,000 acres of a privately owned land, owned by the owners of Subway sandwiches. And they decide to give a big chunk, 30,000 acres, to University of Florida with the criteria that they cannot build sky risers and malls on that plot of land. It's an experimental site. Go out there, take your students out there and work on these systems. It's just at the intersection of Eha Junction. They're going north on the turnpike, 441 and Eha Junction. It's right on the south west quadrant. But it's got this diverse ecotone. It's got almost 40 different types of plant and land covers present in it, right? And that is what gives us the opportunity to go in and evaluate these ecosystems, do some soil health assessment, do some microbial assessment, do some water quality assessment. And then we've got folks like Dr. Smith who can do some digital data repository scaling up as well. But these are pictures that were clicked on our overnight visit there. You can just see the dynamic nature all across one preserve. And so there is potential to not just work with row crops, but to use those same concepts from nutrient conservation point of view, from carbon conservation point of view, back into ecology. Results don't need to go down there. And folks doing, genomic work. We are working with Dr. Mertens and his team out of Fort Lauderdale. And what they do, when they see all these different soil samples, they want to get their hands on it because they want to see if the microbe density and species type are changing. So there's a lot of genomics work these days that folks can do. And I can at least see two scholars sitting here in the room that are doing it, and they showed me their lab. So yeah, this is some of the work that you all can do. And then the hydrologists come into the system, right? And they say, hey, you guys got these wetlands. You got so many isolated wetlands in here. Can we come here and see how these wetlands are expanding and contracting and over seasonal changes to see whether they are stimulating carbon losses as they expand and as they contract. Okay. Now, again, an image I want to just kind of mention here. All the stuff that the professors and faculty that you'll see that we do, it is just the basis. It's the students and the postdocs who are really growing from all these activities and are building these projects, bubbling these projects bigger and bigger. The last few minutes I'm gonna take is trying to highlight the concept of extension. Had a long discussion over lunch. What is extension? What is it that you do in extension? Extension is a science, basically, where we break down our research to a level where our end user, our stakeholder, can understand. If there was a group of third graders outside this room, I would have to go out there and break down what I've been talking with you all to make them understand and stimulate those 7-year-olds to become future farmers, to become future researchers. That is extension, and it's not that simple. It's really not that simple. It's the same with farmers, with the growers. We got to break down our science so that they can touch, they can feel, they can see where the effects lie. And sometimes with research, it's not easy to do that. We get bogged down in our numbers and in our stats. But that's what extension is all about. So it's outreach efforts. Who do we reach out to, right? We've got our stakeholders, we've got regulators, extension agents, we've got the general public. How do we break it down? One-on-one interactions, group interactions. You know, every winter, South Florida Fair attracts 500,000 folks over 17 days. University of Florida, IFAS has a little booth, sustainable agriculture. All the students are talking about ag. We get stragglers come through there and we discuss what we are doing with our community, with our stakeholders one-on-one. So that's what we do with as far as extension goes. We also do some technical things like training the trainers, in-service training, where we develop modules for our agents and we train them so that they can go out and reach out to their end users and their stakeholders. There is also the question, so what? So what if you're doing extension? What does that mean anyways, right? One way to capture that and quantify that is by doing knowledge change or knowledge gain or knowledge change assessment. So we do pre-post assessment to see whether our audience has learned something from it. We do needs assessment to hear from our audience to see what can we do to answer their problems and answer their stripes. So that's what we do as far as extension. And then there's global extension. You can take this at a global, as a matter of fact, it is more prominent at a global level where we take this science to developing countries and try to address their issues. ESP, Epsilon Sigma Phi is one of the oldest extension organizations. And from a professional development, I would encourage every one of you all in the room, look it up, sign up, and try to see if extension is something that you all may be interested in doing. So a lot of things you can do with extension. Last year, we were at in the Caribbean. We were in St. Vincent and Grenadines, talking to a whole host of folks about soil fertility and fertilizer use efficiency. And it was extremely, extremely enjoyable and nourishing to be out there and breaking down that science in their field. With that, I'm going to end a whole host of folks and sponsors that I got to thank. Happy to take any questions. Thank you.